Hello students, in the last lecture we can study the topic that is the cell division. So cells should be divided by two ways, whether the process of mitosis or whether the process of meiosis will occur. In mitosis cell division, from one diploid cell to again two diploid daughter cells are formed with the help of the different stages, and in the process of meiosis, that is the four daughter cells are formed which are haploid in nature, and with the help of the different stages these four daughter cells are formed. That is the major difference in between mitosis and meiosis. That is the formation of the daughter cells. In mitosis two daughter cells are formed which are identical with their parent cell, but in case of meiosis four daughter cells are formed, they are not identical with the parent cell. Suppose again repeatedly I will discuss the mitosis and meiosis. So for the easy understanding of that two concepts, that is the cell division, mitosis and meiosis one, which is the important one, or it is the basic in case of the genetics one. So in case of the mitosis, in last lecture we can study the different stages, and with the help of that different stages we can try to understand how this formation of the two daughter cells which are diploid in nature from the parent cell which are formed one. Suppose this is the figure in case of mitosis and meiosis. Suppose here we can observe it is also called as because of it will be occur in the somatic cell, so it is also called as somatic cell division. So first cell we can observe in that figure is what is the normal cell, and that cell is ready to begin to mitosis, that is the interphase one. So during that interphase, again it will be, that is the longest phase, already we can study in last lecture. It will be divided into three substages, that is the G1, S and G2 phase 1. So when cell is ready to begin to mitosis or duplication of the dead cell, this is the interphase 1. So second importance, that is the stage in case of mitosis, that is the prophase 1. So in second and third figure we can observe that is the in early prophase, coiling of the chromosomes will be started. Coiling, shortening and thickening of chromosomes will be takes place. The nuclear membrane start to be degenerated one. In case of late prophase, we can observe that is the coiling, shortening will be occur. Nuclear membrane which is almost, and nucleolus which is start to be almost, it will be disappearing one. In case of the next stage, that is the metaphase, important stage will be that is in which spindle formation will be takes place, and that is the spindle fiber formation means that is the thread like structures which are developed, and within that particular thread like structure the centromeres are attached to that particular these thread like structures, and we can observe the in case of that metaphase figure these thread like structures are formed one. In case of anaphase, which is the important, that is the in anaphase, in early anaphase, we can observe the division of centromere which is takes place, and once the division of centromere which is takes place, one part of that chromatids or chromosomes which are observed, that is the movement of that chromatids will be start to the opposite poles one. In late anaphase we can observe the clearly division will be takes place, and movement of that one group of the chromatids to the either side of that cell we have to observe one. And in case of the telophase we can observe that is the nuclear membrane and nucleolus which are formed one, and we can observe the two identical groups of the chromatids in case of the telophase one. And in last figure we can observe that two identical groups which are formed, that is the after the development of the nucleolus and nuclear membrane, when the cytokinesis process will be occur. Cytokinesis indicates division of that particular cytoplasm which is occur, and once the division of the cytoplasm which is occur, we can observe the two identical new daughter cells which are diploid in nature, and they are identical with the parent cell one. When we have to repeat this one again with the help of the that figure, this is the explanation of the mitosis. At the base we can observe the S, G1, S and G2 space which are normally occur in case of metaphase first. Sorry, it is occur in case of interphase when cell is ready to begin to mitosis, and after that G2 space, first prophase which is occur in which coiling, shortening and thickening of that chromatids may be takes place. Then in case of prometaphase, that is the initial one, that is the spindle fiber formation which is takes place. In metaphase 1, that is the after the spindle fiber formation takes place, these centromeres are attached to the thread-like structures. Then in case of anaphase, division of the centromere which is takes place, and once the division of centromere which takes place, that will be leads to the movement of the that identical groups of the that chromatids to the opposite side one. And in case of telophase, the nucleolus and nuclear membrane start to be reappear, and we can observe the two identical group of the cells one. And in case of cytokinesis, cytoplasm which is divided one, and two daughter cells are formed in the process of the cytokinesis. So this is the mitosis cell division, in which from one daughter cell, that is the diploid cell, again two daughter cells which are diploid in nature are formed one in the process of the cell division, that is called as a mitosis. So it is also called as a somatic cell division. Which are the important stages? First one is the interphase, second one is the prophase, third one is the metaphase, fourth one is the anaphase, and fifth one is the telophase. And at last, with the help of the cytokinesis, division of the cytoplasm which is occur, and that cells become the separated one. So question will be always asked whether in case of theoretical, what is meant by cell division? Explain the mitosis cell division with the figure 1. So that is the mitosis cell division. In case of meiosis cell division, that is it is a two type of the divisions which are occur. First one is the reductional division, and second one is the equational division. In case of reductional division, which is also called as a meiosis first, and second one is the equational division, which is called as a meiosis second one. So in case of reductional division, from one diploid cell, that is the 2n, two haploid cells are formed in case of the meiosis first, and in case of meiosis second, equational division, from the that two daughter cells which are formed with the help of the reductional division, which are haploid one, equationally dividation of the dead cells which are occur, and from the dead two again four daughter cells are formed at the end of meiosis, that is the haploid in nature one. So in that figure we can consider that is the first normal cell is there, which start to be divided one, or division will be started one. First that is the reductional division which is occur, that is called as meiosis first, in which again there are different stages one: prophase first, metaphase first, anaphase first, and telophase first. So at the end of telophase first, we can observe the two daughter cells which are haploid in nature from diploid because of reductional division. Why it is called as a reductional division? The chromosome number which is reduced from 2n to small n, that is the haploid one. So in case of prophase first, which is the longest phase in case of meiosis. So in case of the that prophase first, again 5 substages are occur one, and which are the changes occur in the that particular that substages we have to study later on one. Then metaphase, anaphase, and telophase. In case of second division, which is the equational division from the that two daughter haploid cells, again metaphase second, anaphase second, and at the end of telophase second, we can observe the four haploid cells which are haploid in nature one. So how this process of the division which is occur, we can study. Suppose in case of the that meiosis, four daughter cells are formed. Suppose the first one, prophase first, that is the important stage one. So in prophase first, 5 substages are occur, in which first is the leptotin or leptonema. With the help of the that figure, we can explain that substage, leptotin or leptonema. So in case of the that changes will be, that is the bead-like structures of the chromosomes we have to observe one. So we can observe that is the chromosomes are appear within the nuclear envelope one. However, they are not fully condensed one. So each chromosome is thin thread-like DNA, and the chromomere structures which are observed one. So bead-like structures of the chromomeres are observed in the case of the leptonin, or it is also called as a leptonema one. So we can observe the green or yellowish colors of the that chromatids or chromonemata, that is the two coil-like structures we have to observe or studied in case of structure of chromosome. So in case of leptonin, which are the changes we have to observe? That is the bead-like structures of the chromosomes which are the observed one in case of leptotin or leptonema. Second substage will be, that is the zygotin in case of prophase first. In case of zygotin, homologous chromosomes are start to be pair with each other, and the pairing of the homologous chromosomes which is called as a synapsis one. So we can observe in that figure this pairing which will be started one, and after the pairing of that structures we can synapsal complex which is observed one under the microscope one. So it is also called as a zygonyma or zygote. Important changes which will be homologous chromosomes start pairing with each other, and synapsal complex which is observed at the end of that zygote one. In case of pachytin 1, that is the two sister chromatids of the each chromosome, they can be separated from each other. And these homologous chromosomes, which are start to be paired one in case of zygote, that is they can be form the four standard structure in case of the pachytin 1. And the process of crossing over, which is occur one. Crossing over indicates exchange of genetic material between that two sister chromatids one. Now, so this crossing over process which is occur in case of the pachytin stage. Then diplotin stage, that is the fourth substage in case of prophase first one. That is at the beginning of this stage, each chromatid of the each chromosome is fused with the chromatid of that homologous chromosome one, and they can be start to be separate from each other one. So this diplotin stage, chiasmata formation which is observed one, and X-shaped structure, that is the chiasma or chiasmata, we have to observe at the end of the diplotin stage one. And last one, fifth substage in case of prophase 1, that is the dikinesis one. So this is the last stage we have to observe in case of prophase first, that is the in which nucleolus disappear and terminalization process will be start to be completed. That is the terminalization is nothing but movement of that chiasmata towards the end, which are occur one, they can become thicker one, and centrosomes are reached to the poles one. So that is the dikinesis substage one. So in case of the meiosis, that is the important one, two divisions are occur. First division is the reductional division, which is also called as a meiosis second, in which first stage will be prophase first. Prophase first is having 5 substages, that is the leptotin, zygotin, pachytin, diplotin and dikinesis. Then next one, that is the metaphase first. After the 5 substages of prophase, metaphase first which is occur. Again we can study in case of mitosis, that is the in case of metaphase, spindle fiber formation which is occur, and after that spindle fiber formation occur, these chromatids are arranged at the center of that particular cell with the help of that thread like structure. Then after the spindle fiber formation, anaphase first will be occur. In case of anaphase, we should know that is the division of the centromere which is occur. Once the division of centromere which is occur, that chromatids may be divided because of the division of the centromere, and we can form four identical groups of that sister chromatids, two at opposite end and two at another end. We have to observe one because of the division of centromere. In case of telophase first, the four identical groups which are formed of that sister chromatids at the opposite end, two one at upper end, two one at lower end, we have to observe one, and with the help of the cytokinesis, once the division will be occur, two identical groups we have to observe in case of telophase first, and cytokinesis will be occur. These two identical groups may be separated one, and here in case of the at the end of the telophase first, we can observe two daughter cells are formed after the division of cytoplasm, but they are haploid in nature. From one diploid cell, two haploid cells are formed in case of the telophase first one. So it is the important division, that is the reductional division, which is also called as a meiosis first one. Then in case of meiosis second, again four stages are there: prophase first, metaphase first, prophase second, metaphase second, anaphase second, and telophase second one. So in case of prophase second, these are the normal cells which are ready to begin to meiosis second one. From that two daughter cells which are equally divided, and there is the formation of the four daughter cells which are haploid in nature. So it is similar type to mitosis one. Here in case of prophase second, coiling, shortening, thickening of the chromatids may be started. In case of metaphase second, again spindle fiber formation takes place, allowing that centromeres at the center, and we can observe the thread-like structure in case of metaphase second one. Then in case of anaphase second, division of the centromere which is takes place, and the movement of that chromatids to the opposite ends we have to observe one. So this movement of the chromatids to the opposite poles of the dead cell we have to observe one. And in case of telophase second, we can observe these four groups, identical groups are observed, which are haploid in nature. And in case of telophase second, nucleolus and nuclear membrane start to be reappear one. And we can observe the four daughter cells at the end of the meiosis second, that is the telophase second, which are haploid in nature. So equally distribution of the that particular or division of the that cells will be occur, and from the two daughter haploid cells, four daughter haploid cells are formed at the end of the meiosis second one or telophase second one. So then what are the significance of the meiosis? That is the majorly we can know, that is in case of sexually reproducing organism, maintenance of the that particularly chromosomes, that is the important one. From the diploid cell, haploid gametes are produced, and when they are fused one, they can also form the diploid cell one. Then in case of the restriction of the multiplication of the chromosome number and maintain the stability of species, meiosis plays important role. Maternal and paternal genes get exchanged during the crossing over. That is because of the crossing over, that is which is occur in Pachetin stage. Exchange of the genetic material from maternal and paternal side, which is takes place, that will be important in case of the variations which are occur in case of the offspring one. Then segregation and formation of the four daughter cells are formed in case of the meiosis. That are the significance of the meiosis one. So it is the nothing but repetition in case of mitosis and meiosis. In last lecture we can also study mitosis, meiosis and their difference one. So in this particular topic we should know what are the difference between mitosis and meiosis, and which are the different stages occur in case of mitosis and which are the different stages occur in case of meiosis one. So important topic in case of the genetics, that is the basic study in case of the genetics one. Okay, thank you.